Thank you for that introduction. And today I would like to give a brief summary of our validation of the genes involved in 9p21 loss and some of the diagnostic and prognostic implications. And I work as a variant scientist at the Molecular Diagnostic Laboratory at MD Anderson, which provides clinical diagnostics for our patients. Let's see. I have nothing to disclose. So the genes involved in 9p21 are CDKN2A, CDKN2B, and MTAP, which are our tumor suppressors. And these genes have wide prognostic, diagnostic, and targetable therapeutic implications. One of the interesting things that we kind of considered as we were going into our validation of the loss of these genes, the copy number loss of these genes, was the difference in the single gene deletion of CDKN2A or B and MTAP. Often these genes do occur together, and the range is quite wide from cancer type to cancer type. And you can see this in glioblastoma, where we have kind of a wider range in the co-occurrence of CDKN2A and B, and then if we look at the lower grade gliomas, we see actually more concordance between those two genes. So we knew it was important to use methods where we could actually see where CDKN2A was deleted and where MTAP was deleted separately. And this presented some challenges. I'd also like to add that this was important for our patient cohort because of the implications of CDKN2A therapeutics, so CDK4 and 6 inhibitors, and then also multiple ongoing trials for MTAP loss with different targetable PRMT5 inhibitors. So this was important for our lab to do. Just a little brief background on our assay. The MD Anderson MAP assay is a hybrid-based capture NGS assay. It was previously validated to detect mutations in over 600 genes, select fusions, and only copy number amplification. So at this point, we were not reporting copy number loss in any genes. We utilized CNV-KIT as our copy number caller in our bioinformatics pipeline. And just starting this year, something exciting that we've been doing is we've been using this in conjunction with precision oncology decision support, which adds annotation and information regarding targeted therapies and the impact of reported alterations. So being able to report copy number loss was really exciting as we were looking at some of these cancers that have pretty important implications. As has been discussed in a wonderful talk about structural variation, there are lots of pluses and minuses to using multiple methods. And there is a slight typo. It's 9p21 loss, and I noticed that last night after I turned this in. First of all, the microarray, some call that the gold standard. Our one limitation was that this had to be conducted in a CLIA lab for our validation purposes, and so we had to find something suitable for that. And then we also had FISH. One of our limitations there is we don't have an exact probe for MTAP. We are reliant on the CDKN2A and B probe, but it is a single cell assay, so we are able to determine ploidy and things like that, and tumor heterogeneity. And then we had external NGS from various biotech companies. And the one limitation with this is that sometimes the validation methods or the copy number thresholds are not defined, or they're proprietary, so it was a little bit difficult to find those. And then some companies only report loss, so we don't know the zygosity of copy number loss or deletion, so they may be reporting loss of heterozygosity or homozygous loss. We weren't exactly sure on some of these, but we did have those available for some of our patients. And then immunohistochemistry, which is useful for the quantification of MTAP, but it only quantifies the secondary product, which is protein, and of course we don't know zygosity or copy number loss with IHC. So our validation procedure started with feasibility studies, where we looked at the patients that we had already sequenced with our assay. And we were able to get a copy number from CNV-KIT, basically showing what we had or what we could determine. And we had pre-existing concordant FISH and concordant external NGS. And then we also had IHC for MTAP, and we were able to kind of look at that and say, can we report loss? Is this something that we can do easily? Is this validation? What is it going to take to validate copy number loss? And then we embarked on a formal validation where we looked at the detection and the thresholding of copy number loss. So what was our cutoff? What was our number that we were actually going to call for homozygous deletions? And so we found some putative homozygous and heterozygous loss samples from our cohort that we had already sampled. We also had fish, and then we sent those samples out for some of those samples out for microarray, and also conducted fish on some of those samples, and then conducted a statistical analysis. After that, we embarked on limit of detection studies where we utilized cancer and normal cell lines with various deletion status. And this was important for determining the threshold for tumor percentage and cellularity. And this is very important in our cohort because we have many tumor samples that are below 40% tumor cellularity. And then we synthesized the metrics and we came up with a definition for reporting. And then something that's ongoing is we're monitoring copy number loss reporting. So we're monitoring when do we call one gene loss? When do we call the triple gene loss? What cancer types are these showing up in? What is our tumor percentage? What does the number look like? So that's something important to us. And then just a few quick insights from our validation. We were able to maximize sensitivity and specificity by determining a threshold for reporting our copy number homozygous loss in 9p21 genes. And we determined that copy number less than 1 met our specifications for reporting accuracy. We learned that somatic CNV NGS detection isn't always straightforward. The copy number value can be affected by tumor percentage, ploidy, tumor heterogeneity, and variation in sequencing. And then also we found that MTAP IHC levels correspond to copy number values, but there is a wide range, and we're looking a little bit further into that now. So if you'd like to learn more about what we did and some of the insights we've gained from our pan-cancer analysis, you can come find me at poster 18 tomorrow to learn more. And with that, I'd like to acknowledge my co-authors, and especially Helen Pelicano, who led the laboratory, the wet lab validation for our project. And thank you very much.